Okay, great. So first off, thanks to Shalev and Arjun for inviting me to this great meeting. My lab specializes in long-term time-lapse microscopy to study cell proliferation at the single cell level. And similar to Arjun, I will also talk about melanoma drug responses. But in this talk, we're going to focus on just the very rapid initial quick responses that cells, melanoma cells, undergo in response to BRAF inhibition, and how cells can actually escape from drug in just a matter of a few days in a non-genetic fashion. So, of course, proliferation is driven by the MAP kinase pathway, RAS, RAF, MEK, and ERK, which drives cell cycle entry. And over 50% of melanomas are driven by an activating mutation in the BRAF kinase, which drives hyperactive cell proliferation. About a decade ago, some great BRAF inhibitors came on the market, which very effectively block proliferation from this mutant oncogene, and they have very dramatic and positive effects in patients, at least initially. But what almost inevitably happens is that some cancer cells find their way around the drug blockade to resume abnormal cell proliferation, leading to drug resistance and tumor relapse. And a major question in the cancer field is, how does this happen? So in one model of acquired drug resistance, an initially drug-sensitive population of cells non-genetically adapts to drug to survive and even proliferate in the presence of drug. And this act of going through the cell cycle and specifically undergoing DNA replication enables some cells to acquire a bona fide drug resistance mutation, which then will lead to a drug-resistant population and tumor relapse. So a lot of effort has been put into sequencing these late-stage relapse tumors to understand the genetic mutations that make them resistant to the drug. But in this project, we are specifically interested in the quick, non-genetic adaptations that cells might undergo just in the first few days of drug treatment, with the idea that this could represent the inception of drug resistance. Okay, so to study these processes, we have heavily invested in new single-cell technologies. So first, we do a lot of sensor design. And in this talk, you'll see a live cell sensor for CDK2 activity, which has really been a gold mine because it allows you to read out proliferation versus quiescence at the single-cell level over time. We also have pushed the limits of long-term time-lapse microscopy, so we can image cells in 96-well plate format with 4 movies per well, with 4 sensors per cell. So these are 4-color movies, and our current record is 12 days of straight imaging and cell tracking, which you'll see later in this talk. Of course, this generates a ton of data, so we've also invested in automated cell tracking, and we have a new cell tracking pipeline that just came out last year called ElipTrack, which is a machine learning-based cell tracking pipeline that is optimized for these high-throughput, multicolor, long-term time-lapse movies of hard-to-track cells. And here's an example of melanoma cells being tracked by ElipTrack. So each nucleus is fit with an ellipse, and this allows us to handle cell-cell overlap, which is a real problem with cancer cells. So altogether, these technologies allow us to do several useful tricks. For example, we can synchronize cell cycles computationally rather than chemically. We can map a fixed cell stain at the end of the movie back to that cell's history to understand the relationship of the past to the present. And of course, we generate the longitudinal data that is really useful, for example, for developing cell lineages. So here's a lineage that goes over 5 days in melanoma cells. So before I show you a movie of the melanoma cells adapting to drug, I want to just quickly go over how the CDK2 activity sensor works, which you'll be seeing. So it consists of a CDK2 substrate tagged with a fluorescent protein. So when CDK2 activity is low, the sensor fluorescence is in the nucleus. But as CDK2 activity builds up, the sensor becomes phosphorylated. This unmasks a nuclear export sequence and masks the nuclear localization sequence, and the sensor translocates to the cytoplasm in proportion to CDK2 activity, which means that in an image, you can read out kinase activity as the cytoplasmic to nuclear ratio of this fluorescent signal. So after mitosis, CDK2 activity turns off in cells that exit the cell cycle or go into G0 or quiescence and starts ramping up as soon as cells commit to the cell cycle. So here on the left is a movie of untreated A375 cells expressing the CDK2 activity sensor, and we're going to follow the cell with the yellow arrow. And you'll see that untreated cells cycle very rapidly. Each red dot is our automated detection of anaphase. And after about 60 hours, you'll see the cells are so overgrown, we just can't track them anymore. On the right are the same melanoma cells treated at time 0 with dabrafenib, which is one of these clinical BRAF inhibitors. So this cell completes the cell cycle it was committed to when drug was added. It divides, and it enters this prolonged quiescence for the next 4 days. And this is exactly what this cell should do because we have cut off its major proliferation pathway. But now I will replay the movie and we will follow a different cell. This one starts off just the same. It divides right around the same time and it similarly enters quiescence. So this cell did respond to the drug. It was not a priori resistant. And it does stay quiescent for some time, but at hour 90 in drug, if you look up at the movie, you'll see CDK2 activity build up and you'll see this cell divide into two daughters right here. So we became, of course, very interested in what was unique about this red cell that managed to escape drug compared to the black well-behaving cell. So what we do is take our many single cell traces and color code them red if CDK2 activity is high and blue if it's low, and then each of these becomes a row in a heat map. So here you're looking at 1,800 single cells that we've tracked for almost 5 days. Drug is added at the black line. And you can see the behaviors are more complex than that initial movie suggested. But in general, you can group the cells into two basic categories. So in the bottom, we have cells that divided once and sometimes twice in drug before entering a long-lived blue quiescence that lasts till the end of the movie. Whereas on the top, we have cells that do respond to the drug initially by entering quiescence, but escape at least once to divide in drug. And when you integrate over 4 days, the fraction of cells that has escaped at least once is actually fairly high, about 43%. So we thought, okay, well, maybe let's just try a higher drug dose and see if we can get rid of these cells. But what we found was that even at this very high dose of dabrafenib, we do reduce the SKP population down to 10%, but we cannot fully eliminate it. We also wanted to test whether perhaps any of these cells had any pre-existing genetic resistance or epigenetic resistance mutations. So to test that, we use this Fuji sensor, which in this case, the cells will light up blue when they're in the cell cycle and not blue when they're out of the cell cycle. So we start off with primarily cycling untreated cells, and then we treat the cells with dabrafenib for 3 days. And most cells go into quiescence, but a few blue ones are still cycling. Then we use flow cytometry to sort the geminin positive, so the blue cycling cells, the SKPs, from the non-SKPs, and plate them down into the dish and give them a 24-hour drug holiday where they can begin proliferating again. And then we're going to treat them a second time. So if the initial ability to escape was driven by pre-existing genetic mutations, then when we sort those SKPs and treat them again, they should proliferate much better than the non-SKPs. But if it's a non-genetic, rapidly reversible effect, then those two populations should look the same. So we filmed the cells, and this is how untreated cells grow pretty rapidly. These are just 3 replicates. And here are cells responding to dabrafenib for the very first time. And then we can look at the sorted SKPs compared to the non-SKPs in red and blue, and we see that they fall all right on top of each other. There's no difference between the SKPs and the non-SKPs upon the second treatment after this drug holiday. So this argues for the ability to escape being reversible upon drug withdrawal and therefore not genetic. So to understand what was driving these SKPs, we use single-cell RNA sequencing. So on the bottom left, you see the untreated cells, and in the top right corner, the treated cells. And A major challenge in our field of single cell biology is mapping between two different techniques. So for example, long-term time-lapse microscopy, where you can track cells over long periods, but you can only measure a few signals in a single cell, versus, of course, single-cell RNA sequencing, where you can measure many, many things, but only at one time point. But in this case, we could map between these two technologies because we knew the SKPs were cycling, and so we can light them up with the cell cycle gene signature and see that they fall in this little pink peninsula. So we wanted to look at genes that were upregulated in SKPs compared to non-SKPs, and in SKPs compared to untreated proliferating cells. And we found, of course, a number of pathways. And I'll just talk about two that we have heavily validated and that we think are very important. And they're actually both stress-related pathways. So the first is the ATF4 integrated stress response. And the second is the p53 pathway. So for anyone not familiar with the integrated stress response, this is a pathway that detects a number of different cell stresses: nutrient limitation, unfolded protein stress. Each stress activates a dedicated kinase, which will phosphorylate a translation initiation factor to suppress global translation, but it will actually activate translation of the ATF4 transcription factor to upregulate stress-related genes. So what we first wanted to check is whether indeed ATF4 protein now is induced by drug. And indeed it is. Here you can see in pink relative to DMSO. And furthermore, when we split this violin into SKPs in orange and non-SKPs in blue, we see an even more significant upregulation of ATF4 and SKPs. So if ATF4 is important, what happens when we knock it down? So here we're looking at the fraction of SKPs, percent of SKPs marked by phospho-RB, and we can see that between control knockdown cells in blue and cells where we've knocked down ATF4, we get about a 50% reduction in cells escaping in the fraction of SKPs. And this actually seems to be largely driven by cell death. So normally we get about 18 to 20% of cells dying at 1 micromolar dabrafenib at 4 days. But when you simultaneously knock down ATF4, that shoots up to about 55%. So from this, we can say that ATF4 is promoting the survival of these SKP cells that are attempting to cycle in the presence of a drug that is trying to block them from cycling, which you can imagine is stressful for cells. We also looked at a number of the top ATF4 target genes. So here's just one, CDC42EP1, which you can see lighting up here in this SKP peninsula. So we validated that by RNA FISH. So the yellow dots here are the mRNA for this gene, and pink marks the cells that are proliferating. And indeed, upon 3 days of BRAF inhibitor treatment, we see a strong upregulation of CDC42EP1 mRNA that is significantly enriched in the SKPs, just as the single-cell RNA sequencing predicted. And then when we knock this gene down, of course, we see fewer mRNAs, but also we see fewer cells escaping. So this is suggesting that not just ATF4, the master regulator, but specific ATF4 target genes are supporting cells that are attempting to cycle in the presence of drug. So could SKPs be the seed population that drive eventual acquisition of drug resistance mutations? Well, for this to be true, they would have to be more likely to acquire mutations, and they would have to outproliferate non-SKPs. So to test the first point, we remembered, of course, that the p53 pathway was upregulated uniquely in the SKPs. So we measured DNA damage using this gamma H2AX antibody as well, and then we also stained cells for DNA in blue and EDU to mark S-phase cells in pink. And we saw very little DNA damage signal in untreated cells, but this signal is upregulated after 3 days of treatment, and it's upregulated fairly specifically in the 3 cells that happen to be escaping and in S phase at that moment. So this is quantified here for thousands of cells, and we can see that the SKPs have higher damage signal compared to the non-SKPs and compared to untreated proliferating cells. We're now trying to understand where this damage is coming from because, of course, BRAF inhibitors are not designed to be mutagenic at all. And one thing we figured out so far is that these cells seem to be undergoing DNA replication stress, which is a result of stalled DNA replication forks, which can be marked by this protein FANCD2. And again, we see low signal in untreated cells, much higher signal in treated cells, and that signal again is enriched specifically in the cells that are escaping at that moment. And it's not just DNA damage marks, but actual DNA breaks, which we can measure with this COMET assay, and we see increasing breaks, fraction of cells with breaks over time. So based on this, we can say that SKPs are prone to DNA damage and therefore will be more likely to acquire over time potentially drug resistance mutations. To test the second point about SKPs outproliferating non-SKPs, we performed a 12-day time-lapse movie with cell tracking, which was a feat only possible because of ELIPTRAC. And we saw that, okay, so first we film the cells for 4 days and classify them before into SKPs and non-SKPs, and then we keep filming. And we saw more proliferation in SKPs than non-SKPs, albeit with long periods of quiescence in between. So these cells don't suddenly become fast cycling once they escape once. They enter more of a slow cycling state. And this is maybe even clearer when we plot each mitosis as a black dot in the two populations, and we see about 3 times more mitoses in the initial drug SKPs. We also looked over longer terms by FACS sorting SKPs in column 3 compared to non-SKPs in column 2 after 3 days of treatment, and followed the same well over 4 weeks. And again, we saw higher proliferation in the SKPs. So this checks the second box as well, that SKPs outproliferate non-SKPs over extended drug treatment, and suggests they indeed could be driving eventual drug resistance. So one last point is that this is not specific to A375 cells treated with dabrafenib. So here I'm showing you several other BRAF inhibitors in yellow and green, MEK inhibitor in blue, and even a combination dabrafenib and trametinib, which is used clinically to stave off drug resistance. And indeed, when we use the combination, we do see a reduced fraction of cells that are still cycling after 96 hours of treatment, but we cannot eliminate this population. This is true for other melanoma cell lines like WM278, as well as for short-term cultures of ex vivo patient biopsies represented here by these 2 MB cells. So in all cases, no matter how high of a drug dose we use and no matter what cell line or patient biopsy we use, we can never eliminate all the cycling cells at any time point. So to summarize, I told you about ELIPTRAC, this new cell tracking pipeline for long-term live cell microscopy. If anyone is interested, we're happy to help you get started with it. With this pipeline, we were able to find that subsets of melanoma cells can rapidly rewire to escape MAP kinase pathway inhibition and re-enter the cell cycle in just a matter of a couple of days. These SKPs experience heightened DNA replication stress and DNA damage, yet they outproliferate non-SKPs, suggesting they could be a seed population that enables drug resistance. So a remaining question is just how widespread are these phenomena of rapid drug escape and associated DNA damage? Could this be happening for multiple MAP kinase pathway inhibitors in multiple different types of cancer? If so, I think this could inform the way the field thinks about the origins of drug resistance and about how MAP kinase pathway inhibitors are achieving their therapeutic effect. And these are questions we're interested to follow up on. So in closing, I'd like to thank Chen Yang, who's pictured right here, who led this project initially, and then she was later joined by Chengzhe Chan, who also worked on, was the lead author on the ELIPTRAC work. Tim Hoffman then joined, as well as undergrad Nikki Jacobson. I'd like to thank funding from the Searle, Beckman, and Kimmel Foundations for this work. And I'd also like to note that we have an open postdoc position for just the type of person who would attend this meeting. And we also have 2 new grants to fund follow-ups to this work. So if you're interested, please shoot me an email, or I think there might be some more one-on-one sessions later on today if you want to talk one-on-one today. So thanks a lot. Happy to take questions.
Thanks a lot, Sabrina, for this wonderful talk. There's a question here from Siwan Zhang. Is SKP phenotype spatially dependent? Does initial cell density matter?
Yeah, so we look for spatial effects, and in our setup, we did not find them. So we look to see, if neighbors of SKPs are more likely to escape or not. And we did not find that in our setting, but that's not to say in some other setting you wouldn't find that. We do all of our imaging so that the cells never become confluent, because of course confluence will push them out of the cell cycle as well, and then you have a confounding effect. But yeah, if the cells are very packed in and you hit them with drug, they actually do better. So there is some dependency on confluency.
Great. Another question is from Jeremy Purvis. Exciting work. For the cells that escape drug treatment and show increases in CDK activity, do you know whether they have skipped mitosis or whether their daughter cells eventually arrest?
Yeah, so they don't usually skip mitosis. They normally divide into 2 daughters, like the one cell I showed in the movie. If cells are going to die in drug, it is usually in mitosis where they kick the bucket. Of course, mitosis is a pretty challenging process for cells. And the second part of the question was, the daughter cells. So they're, the daughter cells of SKPs, they do, and after escaping and dividing, they do go back into quiescence for some time, but they are much more likely to escape again than a cell that has never escaped, than a non-escapee. So they don't start cycling rapidly, but if you watch them over 12 days, they are more likely to cycle again. And that's why we think they will eventually outgrow the non-escapees, and why they could be this seed population for drug resistance.
Great. Okay, another question from Julia Wang. Very cool work. The SKPs are a very small percentage of the cells. So she wonders how come ATF4 knockdown induces such a dramatic effect in drug response.
So the SKPs, by time-lapse microscopy, so if you just take a snapshot time point, the fraction of SKPs at 3 or 4 days is something maybe around 17%. It depends on drug dose. But the longer you watch them, the more cells have escaped at least once. And those cells, we now have started doing, adapted a live cell sensor for ATF4. And basically, upon drug, all cells induce ATF4 to a kind of low, moderate level. But it seems like the SKPs are the ones that are inducing ATF4 much more strongly. And we're interested very much in the timing of ATF. So does high ATF4 induction follow cell cycle entry? Like, is it a consequence of now struggling through the cell cycle, which is, our hypothesis. So I think the cells are, already partly relying on ATF4 just from the, general stress of drug treatment, but the SKPs really rely on ATF4. So that would be my answer.
Cool. Arjun Raj says, awesome work. To what extent would the rate of mutation itself be increased? Like, how many more mutations do you get per subdivision with, say, a 2-fold increase in DNA damage?
Yeah. So we have not measured rate of mutation. I mean, that is always tricky. We're thinking of using one of those, systems where you have something that's not a dysfunctional GFP that if you repair, if you have one mutation, or you could have a GFP that gets broken with a mutation, what the rate of that is, and kind of incorporate that into our time-lapse microscopy to get a sense of the overall rate of mutation. The other approach is like whole exome sequencing of single cells, but we haven't gone down that route yet. So right now we don't know, but we do see actual breaks by COMET assay. So in a significant fraction of the cells, that's of course the primarily the SKP. So I would say it seems to be significant, but exactly what the number is, we don't know yet.
Okay, let's take one last question from Shauna Cooper. Very cool work. While this was conducted on flat imaging conditions, do you think the SKP behavior are going to be similar in 3D conditions, where cell connections are different and drug levels are different depending on where the cell lives within a tumor?
Yeah, so one direction we'd like to go into is to grow these in a 3D culture. So you can grow melanoma cells in these spheroids. So we've dabbled a bit with that. Of course, the time-lapse imaging becomes much more challenging. Well, especially primarily the cell tracking becomes much more challenging. But with the right fluorescent sensors that turn on an escapee situation, maybe we don't have to track every cell. We can just fax sort at some time point and count the fraction of cycling cells. So yeah, that's something we are moving into in the next phase of this project. But obviously, the 3D situation is very important.